Um, we're going to just wait a minute or so while we get the initial surge of people to come in. Then I will start this immediately. Um, and you should be able to see our various panelists already. All right. I'm getting to a temporary lull. Hello, everybody. I am David Randall, Director of Research at the National Association of Scholars, invisible because I've had surgery and am speaking to you from bed rest. So I'll be an invisible ghost during this as the moderator. It's going to be so much fun. I am delighted to have the final webinar in our, History of American Science, American Inventions webinar series, and that's on the Human Genome Project. Now, I want to first say what was the prompt given for this to our distinguished panelists. And that is one of the most significant accomplishments in the history of human health was achieved with the completion of the Human Genome Project, an international effort to identify, map, and sequence all of the genes of the human genome. How did this project begin? What were some of the key challenges that needed to be overcome? What is the legacy of the Human Genome Project today? Now, I'm delighted to be able to introduce three very distinguished panelists, much of whose published stuff I'm not obviously going to say here. It should be appearing in the chat button below at some point in the near future. But briefly, Dr. Eric Green is the director of the National Human Genome Research Institute at the U.S. National Institutes of Health. And he's been at the Institute for more than 25 years, during which he has served as the Institute's scientific director and, oh golly, an awful lot more of really important stuff. Dr. Aristides Patrinos is a member of the Novim group and a visiting scholar at the New York University Center for Neuroscience. From 2006 to 2020, he worked for Synthetic Genomics Inc. in various roles, including as president, and then just an extraordinarily long, distinguished career in addition, with a leading role in the Human Genome Project and in the genomics revolution. And then Dr. Carlos Bustamante is the founder and CEO of Galatea Bio Inc., and he is also a professor of biomedical data science, genetics, and, by courtesy, biology at Stanford University. And again, the usual extraordinarily long CV. The order I have introduced is, I believe, the order in which they're going to speak. Now, I'm just going to say, roughly 12 to 14 minutes each of them speaks. Then Q&A, questions from you, the audience, into the chat or the Q&A buttons below. I can then pass them on, or they can be read by the panelists, who can also just talk to one another in a happy conversation as well. If your question doesn't get answered, and I don't do them chronologically, I do them in whatever order it seems fun and productive, do not worry. You can send me the questions afterwards, randall@nas.org, and I'll be delighted to forward them to the panelists so they may have the option of answering you. And also, don't worry if you have to leave halfway through. This will be recorded for perpetuity on the National Association of Scholars YouTube website channel, available within 24 hours. So no worries for any cause. And I guess I'd ask Dr. Green, would you be so kind as to go first?
Absolutely, happy to. I'm just sharing my screen while we speak. I think you can see my first slide. I thought it would be valuable for me to go first, and because I actually started my entire career just working on the Human Genome Project, literally on the front line from day one when I was still a trainee. I just graduated medical school with an MD PhD and decided to make a career of this. And I think if we're going to have a good discussion, it'd be helpful for me to give a background with slides, which is what I'm going to do. Then you'll hear from Ari, my good friend Ari Patrinos, who was a funder and an important architect of the Genome Project. And third, you'll hear from Carlos Bustamante, who I think was probably in like grade school or middle school when the Genome Project was going on, but he's built a career on the fruits of the Human Genome Project. So let me tell you the story of the Human Genome Project and try to do it as quickly as I can. It turns out that there are a lot of major things that happened last century that were critically important for setting up the Genome Project. Certainly the discovery of DNA's double helical structure in the 1950s, was instrumental in understanding how DNA was the information molecule of all living systems. But that then led to a series of developments, in particular, an era known as the molecular biology revolution that took place in the 1970s and the 1980s that developed cloning technologies and technologies for reading out the letters in our DNA and other methods that were instrumentally important for rigorously studying DNA. And that gave scientists a toolkit for being able to contemplate the idea of getting to the point of being able to read out all the letters in an organism's DNA. All the letters, all the DNA of an organism is a genome. And the idea of having the tools to actually read all the DNA in an organism's genome got a lot of attention by the mid 1980s. In fact, there really was a drumbeat of discussions leading up to the Human Genome Project, major papers in 84 and 86, but also the founding of the field of genomics, which didn't even get named until 1987, and then a series of scientific strategic visions for carrying out the Human Genome Project, as you can see on the lower row. In 1990, the Human Genome Project began. And here was what was originally envisioned for it. We thought it was going to take 15 years. We were going to first study well-examined organisms with small genomes that were laboratory organisms, and then eventually turn our attention to humans, and we were going to do it in that order. It was also envisioned that in each case, we would organize ourselves, we'd map out the DNA, and then we'd actually read all the letters of the DNA. And we wouldn't start sequencing the human genome until we had all new methods for sequencing DNA. We thought at the time the 1990s methods were going to be too slow and inefficient. And we were going to make generating the sequence of the human genome the signature accomplishment of the Human Genome Project. Let me just tell you the spoilers. I'll give you the alerts. Number one was an overestimate. It only took us 13 years. Number two was maintained. We did first study model organisms and then turned our attention to human. We mostly maintained this idea of map and organize first, then read or sequence second. We did not get all new sequencing methods. We eventually adapted the existing sequencing chemistries for actually sequencing the human genome. And absolutely true that the human genome sequence was the signature accomplishment of the Human Genome Project. Those are sort of like take-home messages. If you ask what other organisms were sequenced, this is the list. It was a yeast, it was a fruit fly and worm as two other animals. It was one other mammal, the mouse, and then of course it was human. These were picked in part because they're studied in the laboratory frequently, but they were also picked because you could see they represent different key points in the evolutionary tree. Now, let me remind you what the world looked like in 1990 when the Human Genome Project launched. First of all, it was not a complete consensus in the scientific community that this was a good idea. Many scientists were opposed to the Human Genome Project. Second, and I was there on day one, there was no start to finish plan of how we were going to actually accomplish the goal. It was one of those figure it out along the way, build the airplane as you're flying it kind of a thing. And for all 13 years, we had to figure it out, and we readapted what we were doing almost every few months. The field was a very young toddler field. It didn't exist until 1987 and really became a melting pot of immigrants from other disciplines. I came from medicine and pathology. Others came from engineering or mathematics. Some came from biology. Some came from genetics. It was painfully early days of a functional internet. People don't realize that when we launched the Genome Project, we needed a functional internet. There was barely a functional internet. In fact, the early ways sequence data was being distributed back when the Genome Project began was initially through books. Eventually, we upgraded to actually working with CD-ROMs. And even when the Genome Project began, we were using fax machines to exchange data. Here's a fax machine that represents what I used in some of the earliest days of the Human Genome Project. It was a very different data science era back when the Genome Project began. How are we going to do the Genome Project? Well, funders from around the world came together. The National Institutes of Health funded the work that I was doing. And in fact, the institute that I now lead was created by the US Congress to lead the US's National Institutes of Health's effort in the Genome Project. Patrinos will tell us about the Department of Energy's role, and he led and was the architect of the Department of Energy's role. But other countries got involved as well. Some of them were government funders, some of them were private funders. It was one big team working on the Human Genome Project, especially when it came to sequencing the human genome. And as we sort of said, the plan was when it came to studying the human genome, we divided and we conquered. we split up the genome and then we first mapped it and then we sequenced it. To put a personal and fine point on this, here's sort of a satellite view of the human genome. This is looking at our 24 different chromosomes, 1 through 22, X and Y. My early career as a trainee working on the Human Genome Project was all focused on chromosome 7, working with my mentor, Maynard Olson, shown here. And here are some photos of me when I was much younger, during the beginning of the Genome Project, in the middle of the Human Genome Project. And on the lower photo, you can see some of the maps of chromosome 7 that we were building prior to sequencing it. Now, how did we figure out what to do? How did we actually organize literally thousands of scientists around the world to work on these different organisms and then work on the Genome Project when we sequenced the human genome? And we did it through this series of strategic visions. And these were sort of documents that came together with the scientific community and that were written up and prominently distributed or published. And there was actually 3 of them, those 3 shown here, that represented guiding blueprints for how to accomplish the goals of the Human Genome Project. The good news is everything seemed to be working throughout the Genome Project. There were moments that it didn't work so well, but by the late 1990s, those model organism genomes were getting sequenced. First the yeast in 97, the worm in 98, the fruit fly in 2000. And so by the late 1990s, about 7 or 8 years into the Genome Project, it came time to start sequencing the human genome. We had learned how to sequence genomes by studying model organisms. Now it was time to turn our attention to the Herculean task of reading out the 3 billion letters, or nucleotides as they're called, or bases, 3 billion of them that make up the human genome. We were going to use the methods that were available at the time and that we refined, but they gave very short bits of sequence at a time. That made it very difficult, which meant we had to read every letter of the human genome many, many times. At the end of the day, we read every letter of the human genome at least 30 times to convince ourselves that it was going to be highly accurate when we said it was a G or an A or a T or a C at that position. And I would also say this was made much more difficult to put this all together because you may not realize, if you're not a biologist, that about half the human genome is highly repetitive. And so it looks like pieces that you see everywhere else. So sometimes you wouldn't know when you had a sentence in the human genome, whether it belonged on chromosome 3 or chromosome 10, or maybe it appeared 25 times on chromosome 7, and you had to figure out which place it resided. So it was like pushing a boulder up a mountain. It was incredibly difficult. But at the end of the day, we were able to do it. And we did it because we automated everything. We had incredible technology, such as these machines that are shown here in a factory-like form that sequence DNA very efficiently. And we eventually got the kind of computational power that was going to be needed for developing that first complete sequence of the 3 billion letters in the human genome. Here are some numbers you might be curious to know. At the end of the day, 6 countries were involved in sequencing the human genome, involving 20 centers and thousands of researchers. While we were sequencing the human genome, we were generating 1,000 letters or 1,000 bases of data every second, 24 hours a day, 7 days a week for 6 years. And it was really this brute force sequencing and massive computational help that allowed us to get the job done. Some of you might have heard about or read about the human drama that came in with the Human Genome Project. There was human drama. It was a sociology experiment on top of a scientific experiment. The 2 major protagonists that you've read about, or maybe Ari and I will comment on, or maybe Carlos from his perspective, was Francis Collins, who was the director of the institute that I now lead. But he was leading the NIH's effort, and in some ways he was help leading the whole international Human Genome Project effort. And then there was this other scientist, Craig Venter, who was originally working on the Genome Project, but then went off into the private sector and started developing work in a company that tried to compete with the Human Genome Project. I'm going to leave the drama behind. You've probably read about it, but it does explain why, when the Human Genome Project went to publish the draft sequence of the human genome, there was actually 2 papers that appeared. One appeared in Nature, which was the Human Genome Project's paper, but then Craig Venter and this company, Celera Genomics, they published their working draft sequence of the human genome in Science. And it was declared a tie at that point, and most of that drama then melted away for the last few years of the Genome Project because the Human Genome Project went on to complete the task. In April of 2003, the Human Genome Project came to an end officially. All the goals had been accomplished. We didn't need 15 years. And for those who follow some of these things, Congress got in the act and they declared National DNA Day to be April 25th of every single year as National DNA Day to commemorate the anniversaries of DNA's double helical structure coupled with the completion of the Human Genome Project. And to this day, we still celebrate National DNA Day every April 25th. So the highlight features of the Genome Project, it was completed ahead of schedule and under budget. We generated an incredibly highly accurate sequence for about a little over 90% of the human genome. It wasn't totally complete. It was nearly complete or essentially complete. I'll say something about that in a second. The cost was about $1 billion to generate that first sequence of the human genome. That's a lot of money. We've now reduced that over 1 million-fold, but it was an expensive thing to do the first time, but it was incredibly important. That race I was telling you about between the Genome Project and Craig Venter sort of melted away after 2000. And similarly, I would point out that the initial concerns about the Human Genome Project from some parts of the community have melted away. I routinely talk to senior colleagues in the community who say, I was once against the Genome Project, but you won me over. It was one of the smartest things we ever did. And meanwhile, the Human Genome Project set the field of genomics into a trajectory of widespread dissemination across biology and medicine and society. And one other thing I did want to say in terms of an epilogue is that some of you may have heard last year or this year about the sequence of the human genome being finished again. I want to make sure you understand the difference. The Genome Project got really high quality sequence for 92% of the genome. But that remaining 8% was not readable at the time 20 years ago when the Genome Project ended. And there were big problems with those regions using the methods that were available. The good news is we now have thrown out all those old methods and we have all new revolutionary methods for sequencing DNA. And these methods, plus better computational approaches, set the stage for a whole new group of researchers, younger than any of us on this panel, who finally have gone through and figured out that last 8%, so that we have a truly complete sequence of a female human genome in 2022. And then earlier this year, just a few weeks ago, was announced that they actually got a complete sequence of the human Y chromosome. To put it in perspective, I sometimes like to make analogies to the moonshot. I'll remind you, the moonshot was one of the biggest scientific endeavors of my lifetime. It was expensive, $25 billion, and it was highly managed. And the signature goal, put a person on the moon, has been repeated 11 times since the moonshot. The Genome Project was biology's equivalent of the moonshot. It really did represent an important biological managed project. It was much cheaper than the moonshot, but still, it was a lot of money. In total, it was $3 billion for the whole project. But what I think is remarkable, in the last 20 years, after sequencing that first human genome, our signature goal has been repeated more than 1 million times. The moonshot repeated 11 times, the genome sequence, human genome sequence generated over 1 million times. I think we can be very proud of that. So that this year, when we went to celebrate the 20th anniversary of the Human Genome Project, a lot got written. I'll just give you on my last slide. I just like this quote. It came from a magazine outside the scientific community, The Economist, who wrote this year, about 20 years after the Human Genome Project, how the project really revolutionized biology. And I just want to leave you with this quote, that they said that no part of biology is untouched by the consequences of the Genome Project. Genomics has therefore come to form a framework for biology in the way that the periodic table forms one for chemistry. It touches everything. And with that, I'm going to stop, and I'll stop sharing my slide, and I'm going to turn it over to my good friend Ari Patrinos, who I'm sure will add some commentary to what I just outlined in terms of the history. Thank you very much.
Thank you very much, Eric. That was a wonderful summary, and I plan to mostly cover how the Human Genome Project, in fact, got started, because I think putting it in that context may be interesting, at least to some of our audience. A little bit about myself. My entire career, including some forays in the private sector and academia, as you've heard, has been associated with the U.S. Department of Energy and several of its national laboratories. For example, Oak Ridge National Laboratory, Lawrence Berkeley National Laboratory, Los Alamos and Livermore, including Argonne when I was a graduate student, in fact, at Northwestern. For those not knowledgeable about the Department of Energy, it's the successor to the Atomic Energy Commission that was created right after World War II, and specifically after the bombs at Hiroshima and Nagasaki, the so-called dawn of the atomic era. If you've seen the recent movie, you may have some ideas about that. In addition to the excitement about energy too cheap to meter, which was the kind of lingo back then, was both the horror, of course, of mass killings, as well as the realization that even releases of low-dose ionizing radiation may have deleterious effects on biology, and especially human biology. The low-dose radiation concern was, in fact, behind the setting up of a division of biology and medicine within the Atomic Energy Commission, primarily to coordinate biomedical research involving atomic energy in 1947. Then I remember that date because it was, in fact, the date of my birth. They also set up an advisory committee for this research that reported directly to the Atomic Energy Commission chairman. The Division of Biology and Medicine eventually became the Biological and Environmental Research Program that I've had the privilege of leading between 1993 and 2006, with various other jobs with the DOE and its national laboratories before and after that period. I have given you this bit of history not because of any nostalgia, but rather to tell you how the Human Genome Project got started. Starting in 1947, decades of biological research by the Atomic Energy Commission and DOE were devoted to determining low-dose ionizing effects on mostly human biology. Thousands of rats and other creatures were irradiated at multiple national laboratories. And yes, even human beings back then served as guinea pigs, mostly volunteers among the terminally ill cancer patients. Needless to say, very little progress was made during those decades, primarily because of the rather primitive tools of biomedical research. Genetics discoveries around the middle of the 20th century prompted the recognition that DNA research may prove the way to pin down these effects of ionizing radiation, along with other obvious applications and breakthroughs in medicine and health research. So it was, as they say, that in the early 1980s, my predecessor, Charles DeLisi, after stints at Los Alamos National Laboratory and the NIH, found himself heading the BER program in the Department of Energy. At that time, it was called the Office of Health and Environmental Research, existing within the Directorship of Energy Research and surrounded by the behemoth offices such as high energy and nuclear physics, fusion energy, chemistry, high performance computing, and the like. In 1986, DeLisi convened a workshop in Santa Fe, New Mexico, to consider launching an ambitious research program to sequence the human genome. The workshop deliberations produced a fairly detailed plan that was incredibly prescient in terms of detailed activities, timing, and costs. It even predicted the increased involvement of the private sector, forecasting in some sense the emergence of cellular genomics. The price tag of $3 billion was most probably, I would say, a simple multiplication of 3 billion base pairs times $1 per base pair. Many stars aligned that led to the strong support for the Human Genome Project within the Department of Energy. The director of the Energy Research Directorate, the boss of DeLisi, basically, his name was Al Trivelpiece, together with the Undersecretary of the Department of Energy, Joe Salgado, were sold on the timing and the value of this research undertaking. Also playing a major role, maybe the major role, was Republican Senator Pete Domenici of New Mexico, who held important purse strings and enthusiastically endorsed the projects. He also had 2 national laboratories within his state, which was another impetus for his support. My NIH colleagues were not exactly thrilled by the DOE goings-on. Some of them thought the project was premature, awaiting advances in the sequencing technologies. Others because they thought only 3% of the sequence was valuable, the genes basically, and the rest was just junk DNA. And perhaps most importantly, they felt that such a project should be undertaken and in fact led by the NIH. More stars started aligning as the National Academy of Sciences convened a study under Bruce Alberts and strongly endorsed the Human Genome Project, also joined by the Congressional Office of Technology Assessment, the Office of OTA, as it's known, that had a similar conclusion. OTA was in fact the one that Mr. Newt Gingrich eliminated, arguing that Congress didn't need any science or technology. I leave that without a comment. So it came to pass that DOE and NIH were basically told to play nice with each other and to jointly administer, the Human Genome Project. Thus, with all the predictable fanfare, the Human Genome Project was launched in 1990 with a vague horizon of 15 years and an even more vague budget of $3 billion. And about that, you've also heard from Eric a little while ago. So this is the story of how the project began. Or is it really? There is another side to it that I want to discuss during the remainder of my time. Allow me to bring in one of my greatest heroes, the physicist Freeman Dyson, that I was honored to know and to learn from. He speaks about two types of scientific revolutions. It's in his book, Imagined Worlds, the concept-driven one and the tool-driven one. The first seeks to explain all things in new ways. The latter discovers new things that need to be explained. We have generally had much better luck and much more fun with tool-driven scientific revolutions. Astronomy epitomizes a tool-driven scientific revolution, as when we build a bigger and better telescope and we turn it towards the heavens, entirely new things are revealed, and we have fun trying to explain them. As I said earlier, my office within DOE was squeezed between the behemoths of physics, chemistry, fusion, high-performance computing. For over a century, such disciplines enjoyed the lion's share of support, in fact, still do to some extent. Mostly in terms of scientific research facilities, such as light sources, neutron sources, accelerators, and so on. These were and are huge machines costing billions of dollars, dedicated to such things such as smashing particles together in order to figure out what they're made of and to understand fundamental nature of the universe. It turns out some of those facilities also have helped biology in terms of determining the 3D structure of proteins. Also, conventional wisdom also assumed that related knowledge would also lead to better weapons for our national defense. The way federal R&D funding works is you split the dollars between what covers the basic R&D of thinking and doing, and that of using tools, referred to usually as scientific user facilities, emphasis on the user. And you may be able to nickel and dime the thinking costs, but for user facilities, the imagination runs wild. This is what happened with the DOE proposal to sequence the human genome. In some sense, we managed to convince the powers that be that our effort was a tool-driven scientific revolution that had similarities with the big toys that physicists and chemists were playing with. As a result, we deserved the levels of funding enjoyed by these physicists, chemists, high-performance computing nerds. If there is one thing that I am most proud of accomplishing is by hook and by crook, creating 3 major scientific user facilities during my time leading the biological and environmental research program. One of them is familiar to most of you, the Joint Genome Institute. That was essentially the DOE contribution to the Human Genome Project of the combined agencies, the Environmental Molecular Sciences Laboratory, and the Atmospheric Radiation Program. This last one is in fact part of the global climate change research effort that we managed to label as having several scientific user facilities, and therefore being in the same funding category as some of the more established and well-known scientific user facilities. I am not criticizing in any way, shape, or form the hypothesis-driven model of research that NIH mostly follows. In fact, it is the foundation of how advances in R&D are accomplished. It also complements the primarily tool-driven research that we pursued in the Department of Energy. Perhaps appropriately, after I left DOE, all human biology R&D within it was terminated with the emphasis on the biology of the remaining life forms, such as microbes, plants, and others, with applications to energy and environmental cleanup, for example. There are a couple of exceptions, such as two programs, the low-dose radiation effort that's currently been revived. So they expect the DOE to be involved in that, and that involves human biology. And incidentally, also the National Institutes of Health are not particularly interested in examining that part of research because of all its political implications. And also the cancer moonshot involving also the Veterans Administration, something that in recent years was started by the current president, President Biden, and that got also DOE and me back to DOE for a short time. So those are my thoughts about how we started, how we developed it, and what are the sides to it, including this business of the user facilities and how they helped launch scientific programs and research that could compete successfully with the behemoths of the science, such as physics, chemistry, and the like. Thank you for listening to me.
Thank you so much. Dr. Bustamante.
Thank you so much. First of all, I just want to say it's such a privilege and a pleasure to share this stage with Eric and Ari, two dear friends and people I've learned a ton from. So for me, I want to kind of leave you with my message and then tell you how I got here. And that message is that to me, the greatest fruit of the Human Genome Project is how it revealed our common genetic origins in humanity, or a pluribus unum, out of many one. And this was always kind of the underlying logic behind the program, right? We got to get to the human genome. What does it mean to sequence a human genome? Understand the roadmap that makes up all the genes and regulatory elements of the human genome. But at the end of the day, because we are a beautifully diverse phenotypic and genotypic species, it's about the diversity of the human genome. And that's been kind of the story of my career, my interaction with the Human Genome Project. I wouldn't have had a career were it not for the Human Genome Project. I entered the field, as Eric was saying, in 1994. I started working in the laboratory of Richard Lewontin, who was a critic of the Human Genome Project, to be fair. And then I started my PhD in 1997. And when I started my PhD, the Human Genome Project was still a decade out. And I remember people telling me there's no place that's going to want two people that study the statistics of DNA. That's a very niche field. And it felt a lot like 19th century physics. We've got it all worked out. We just need to figure out this black body radiation problem. And the whole field exploded. So I've had the privilege of a 20-year plus career where I've seen a lot of the dogmas that we thought we understood about human origins and human genetic diversity upturned, specifically because we had the Human Genome Project. So to me, I've been a population geneticist my entire career. It's what I've been fascinated by. And had it not been for the Human Genome Project, most of what we've learned over the last 20 years just simply wouldn't have happened. So we've really rewritten our understanding of how humans came to be. as many of you know, Svante won the Nobel Prize 2 years ago for his contributions to sequencing the Neanderthal genome. That wouldn't have happened without the Human Genome Project. So this is just a one representation of our current model of human origins and how understanding human genetic variation is at the core of this program. And I'll begin with one of the slides that we were able to build early on in this process. So this is based on data collected by a company called GlaxoSmithKline. So GSK had genotyped a bunch of individuals in one of the largest studies to date of human genetic variation. This came out as a result of the next phase of the Human Genome Project, which was the SNP projects, HatMap, and from HatMap we began developing arrays so that we could do large-scale genotyping. And the very big picture that came out is that it broke down any traditional notions of race that you may have. There aren't 3, 5, 10 human races. That's just an inappropriate model. There are clusters of human genetic variation, and there are clines of human genetic variation, and there are individuals of so-called admixed ancestry, and that diversity is revealed automagically when you put the genotype matrix of an individual sample into context. And so we can see aspects of history written in the patterns of genetic variation through the analysis of statistical models. So this could be admixture in individuals of Mexican ancestry. It could be diversity within East Asia. It could be clines within South Asia. Probably what made me Instagram famous was this figure where we looked at the first two principal components of the genotype matrix for individuals within Europe. And we came to this cheeky conclusion that genes mirror geography within Europe, and that you could actually take an individual, project them out onto these principal components and actually get a physical representation of where they also live in that genetic ancestry space. And that's now become a multi-billion-dollar business through the launch of Ancestry.com's Ancestry DNA business, where I was an advisor, 23andMe, MyHeritage. Everybody and their mom now has an Ancestry genetic company that's aiming to do this. But the reality is that none of this would have happened without the Human Genome Project, the placing of these markers, the ability to do large-scale genetic analysis for dirt cheap compared to what it cost before we did the Human Genome Project. So there's a hundreds of billions of dollars return on that initial investment that today have spurred on one of the most interesting parts of the US and global economy. And this is only the scratching the surface. As we know, most of human genetic variation stems from our common origin in Africa. So you can apply these technologies to understand population structure in Africa. This is work we did years ago with Sarah Tishkoff and others. Individuals of African admixture in the United States, African Americans who trace their ancestry to both European and African ancestry. We can deconvolve. We can get to so-called chromosome paintings, where we're able to look at stretches of ancestry in individuals of African ancestry, recent admixture, individuals who are self-described as African Americans who mostly have European ancestry. So the entire richness of self-described ancestry compared to genetic ancestry, compared to what's relevant, sort of has something that's been, a beautiful side product of our understanding of human genetic variation. We were also privileged to be parts of projects like the 1,000 Genomes Project and other efforts to collect samples from throughout the Americas, both admixed individuals as well as individuals of Native American ancestry. So this was work we did with the University of Miami years ago, again, using commercial arrays. This is data from the 1,000 Genomes Project, where initially the project was going to have samples from Europe, Africa, and Asia, and we were able to convince our colleagues that it was important to study admixture, bring in populations from the Americas, and today these populations are used as a common resource. Cell lines were made. We did the sampling in Colombia, Mexico, Puerto Rico. This is just one example of how you can study and reconstruct patterns of Native American migration predating European colonization and contact, based on these public resources that are now part of the fabric of how the community operates and carries out research. Finally, we were able to do some just fascinating work on origins of the most remote places in the world. These are people from Easter Island, one of the last places to be colonized, and had a series of papers most recently on the origins of Easter Island and demonstrating that it was founded by populations of individuals of admixed ancestry from Native American and Polynesia, predating European contact. Again, none of this would have been feasible based on the reagents and the way we were creating these reagents at the time when the Human Genome Project was completed, but that on an inordinate amount of fascinating work. And one of the coolest things that we were able to demonstrate is that even trait mapping really benefited from the production of these reagents. So this is a set of beautiful kids from the Solomon Islands that you see. They have this striking blonde phenotype. And as you know, today, there have now been thousands of studies on the genetics of complex traits carried out. This is one of my favorites that we did on the genetics of blonde hair. And we were able to demonstrate there's a massive hit. Some of you know this is called a Manhattan plot. We like to call this our Dubai plot, where we found that the genes underlying blonde hair in Melanesia are the genes that underlie a form of albinism in South Africa, but they're different from the genes that underlie blonde hair in Europe, which is, as we might call this, chemically induced blonde hair variation. But we know a lot about the genetics of blonde hair in Europe. And that's part of a theme that I'd like to close with, which is that there's still ample opportunity to contribute to this. Today, genetics forms the basis by which we de-risk drug targets. PCSK9 inhibitors now are the next generation of therapies for lowering cholesterol. This came out directly from the genetic mapping of families with high cholesterol. We were then able to find protective loss of function mutations. This is now part and parcel for how companies like Regeneron regenerate and create new drugs for us. Resources like the UK Biobank have contributed to this. So the UK Biobank just released 500,000 genomes, one of the largest collections of genetic variation data amenable for this kind of scientific dissection. Again, a complete result of investment by the US and other governments in the creation of the original Human Genome Project, but then spurred on additional investment. It's not all roses. We still live in a world where there's ample underrepresentation of certain groups. So more than a decade ago, we began calling attention to this issue. At the time, about 96% of the GWAS studies were populations of non-European descent. It's gotten somewhat better, but not that much better, to be frank. So this is a recent paper from Alicia Martin, who was a postdoc at the Broad and was a student with me and is now at Mass General leading her own group. This shows the growth of studies done in European population, some increase in populations of East Asian ancestry, but we're certainly not doing nearly enough on a global scale. And this has consequences. We don't really do as well in predicting phenotype in non-European populations as we do in other populations, and we need to do better. And I think similar to what we've heard about, this is not just something we can expect the government to do. There's plenty of opportunity here for private capital to step in and play a role. So this is part of why we founded Galatea Bio, which is looking to catalyze on the once-in-a-generation opportunity we've had to take materials that were used as part of the COVID surge and repurpose them for genome sequencing. So we did a pilot project at Stanford where we were able to sequence 2,000 patients that had been seen in clinic, link that data, link it to the electronic health record, identify genetics of COVID susceptibility. And today, this is just one of the examples from that figure. Today, we're now sitting on a much better understanding of the genetics of a trait that didn't even exist, which was post-COVID infection 2 years ago. We now understand this better than we understand diabetes or heart disease because we've been able to digitize phenotypes and get ahead of this. It's still early days in this, and I'll show you what this could look like. So our goal is to get to about 10 million samples that are from underrepresented groups tied to electronic health record data that allow us to power an understanding of the genetics of health and disease. We're doing this focused largely in the Americas, and I'll show you this. Our goal is to build the largest, most diverse phenotypic database out there, all building on the success of the Human Genome Project. And hopefully this will be my last slide. This is what this currently looks like So we're sitting on about 500,000 samples in warehouses down in Miami, Florida. We're currently processing this material to get to a large-scale understanding of the genetics of many traits, not just COVID susceptibility, but many people who had COVID also obviously had cancer before they had COVID. They're developing heart disease. It's just one of many materials that can be used and repurposed for this kind of work. So with that, I'd like to close and just say that we're looking at a world where ancestry is going to play a bigger and bigger role. We've been able to demonstrate the value in these materials. This is an example of the kinds of diversity in these sets of samples, and we think the future is bright if we can continue to build as a community on opportunities and move forward on a polytheistic approach to generating genome-phenome databases. Okay, with that, I will stop my sharing and turn things over to the discussion. Thank you, everybody.
And thank you so much. I'm going to encourage people in the audience, please do start to put in questions. I'm going to start with one of my own. How many people worked on the Human Genome Project, and to what extent was experience in the Human Genome Project important in remaking the new disciplines coming out of it?
So I could try. I think I passed it by telling you I'm not sure I have an exact number. I think we typically tossed around a couple, if you sort of add in all of the people that were technicians and computer scientists and bioethicists and biologists and geneticists, I've often heard numbers like a couple thousand thrown out there over the course of the project, maybe, sort of in the 2,000 to 3,000 range. So that would be my estimate. My colleagues can try to, correct me if they think it's way off. In terms of the second question, if I understood, was more like, was it really an important thing to work on the Genome Project to benefit from, or to sort of carry it to the next phase? And what I would say is, I think you saw both. There's a lot of people that worked on the Human Genome Project that then went off and don't even work in genomics anymore. There are a lot of people like me who have carried it forward in certain areas. I did it more towards medicine. I think some of the people in the DOE use it to do things that are of interest in agriculture and in biofuel development and in remediation and various other areas. I think the microbiologists have gone totally crazy with it because the genomics has an ability to analyze microbes with their very small genomes in unprecedented fashion. And then there's people like Carlos, who didn't work on the Genome Project at all, but totally took the baton from those of us who did work on it and has built a career on it, a very fascinating multiple areas of great interest. And there's a lot of people like Carlos out there who have picked up the baton and run in many different directions. In fact, one point I'd want to make is that once upon a time, if you just take the NIH as one example, where we have represented many different aspects of biomedicine. But, if I go back 20 years when the Genome Project ended, very little genomics research was being done by any institute except the National Human Genome Research Institute. And now, 20 years later, we're doing only about 10% of genomics research at NIH. The other 26 institutes do 90% or more, because now everybody doing biomedical research touches genomics in some meaningful way.
Does anybody else want to answer that question?
Well, I would just expand a little bit on what Eric said with respect to the applications of genomics to such things as clean energy, carbon sequestration. In fact, ultimately the impact with respect to the survival of this planet may very much depend on how successful we are in those fields. There is some hope. Another example is some way by which we could change photosynthesis so that it wouldn't be as inefficient as it currently is, and at the same time, if we are successful, would solve both the energy problem as well as the food problem. So I think genomics has given us so many additional opportunities for improving the lot of humanity.
That's like why the reason I ended with the quote from The Economist, where they said genomics has sort of come to form the moral equivalent of the periodic table of chemistry. It touches everything. There's almost impossible to touch anything in life science, anywhere in life science, without genomics being all over it.
Dr.
Yeah, I mean,
Bustamante?
yeah, no, I would say the current biotechnology industry is a direct result of the genomics revolution. I mean, it really is a very transformative um, uh, set of reagents and approaches that were unleashed. And you're talking about $100 billion a year market just in financing of genomics operations. So it's massive, and hundreds of thousands of people today, I think, are in this industry as a direct result of the sequencing of the Human Genome Project.
And does this answer actually Vicky Galbraith's question? Does direct-to-consumer DNA testing have any benefits to the scientific community, or is it just useful for entertainment purposes for the general public? Is this a slightly different question to answer, or did your previous answer cover that?
No,
Slightly
no,
different. Go ahead, Carlos, you start, then I'll add to it.
no, I would say that first of all, most of what we know about the genetics of complex traits can be traced to three sources. One is a company called Decode Genetics that got started in Iceland before the Human Genome Project and then accelerated after the Human Genome Project. Just in terms of like papers in Nature Genetics, right? Decode would have a paper, the US Broad would have a paper, and then the UK would have a paper. So I would say absolutely there wouldn't be an understanding of the genetics of complex traits if it wasn't for those efforts. And the latest entrants into that have been 23andMe and even Ancestry.com contributed during the COVID surge in sharing data. So we've certainly understood a lot about the genetics of complex traits by the sheer number of people who've been involved in commercial genetic testing that includes direct to consumer, but also in labs. Natera has sequenced 10 million samples. They announced this, a few months ago. Think about everybody that Myriad has sequenced, GeneDx. I mean, it's massive numbers.
And so I agree with everything Carl said. I'll just add one other subtlety to this. One of the things we think a lot about at the National Institutes of Health is preparing the general public for genomics finding its way into mainstream medicine. And when that's going to happen, one key aspect of it is to raise the genomic literacy of the general public, getting them interested in learning just the bare basics of genomics so that when their healthcare professional talks to them about their genomic findings, they'll understand what's being explained to them. I find it that the direct-to-consumer DNA testing market has been helpful in a playful and many times recreational way through Ancestry and some other things. I think it's gotten people very interested so that they're talking about it. Nothing pleases me more, to be honest with you, than when and this happened as recently as like 3 nights ago, I'm watching CNN at home, and there's an advertisement about 23andMe talking about genomics and your DNA and this and how. And to me, it's right in their living room. I can't afford at the NIH to pay for such advertisement. And the fact that they're getting genomics being discussed in the living rooms of Americans through advertisements on CNN might just capture interest in genomics so that these people are motivated to learn. And I really want society to get excited about genomics because I so much believe it's going to be part of their medical future. And so to me, that's another aspect of it, is to create interest and excitement about genomics. And I think the direct-to-consumer market has been helpful in that regard.
So this is the time to be a black pill. So let us say that what genomics is really doing is making possible targeted diseases for individual genetic groups, and that this will be first seen when a strange disease strikes the Uyghurs, with say, a COVID plus tied to the Uyghur genetic, particular range. And so, what is being done? So how should I put this? Are you at all afraid of the black pill possibilities of the weaponization of this, and what is being done by the government to ward off such things?
I guess you've asked about the government. I'm the government guy. I should answer it. So first thing I'd say is whenever you build these scenarios, they're oftentimes biologically not realistic. I mean, so this whole thing about targeting, populations, I mean, Carlos could be much more sophisticated than I could. that these scenarios that get built don't have as much scientific plausibility as you think they do. But let's just say, for the purposes of discussion, do I think there could be bad things done with genomics? Yeah, sure, I do. There's a lot of nefarious things one could imagine doing, as is true with every other technology humankind has ever invented. There's always a negative thing that technology can be applied for. I am very proud to point out, and I didn't point it out in my talk earlier because I just had to keep it very abbreviated. One of the distinguishing features of the field of genomics, which started on day one of the Human Genome Project, was the dedication of a portion of the budget, which continues to the present time, where I dedicate 5% of all the research dollars I spend at my institute and the government, we spend on studying the ethical, legal, and social implications of what we do. It is somewhat unprecedented, but the field has absolutely embraced the idea of, in parallel with scientific advances, studying what are the ethical, legal, and social implications of those advances. And that has helped us along the way sort of defray possible nefarious consequences of genomics, including genetic discrimination, including issues around genetic privacy, et cetera, et cetera. Are there societal challenges? Absolutely. It's true for all medical advances, all technological advances. But I think as a field, we've been incredibly responsible, and negative consequences I don't think are very demonstrable, at least up to the present time. Carlos, do you want to add to that?
Yeah, no, I would agree with you. I think this is a little bit of fear mongering, and I would say the most important thing to remember is that most variations within and not among groups. So it's very hard to target genetically anything like this, and unfortunately, you don't need genetics to target people. You just need ignorance, and I would say let's try to keep those out of the discussion. My hope is that what we'll see are more on the other side. We just had the FDA approve the first genetic CRISPR therapies for sickle cell, right? That's a win. That's a win for communities of color. That's a win for bringing precision health at scale to everybody. So, the future is bright and good, and it is the prophylactic deployment of medication that will avert disease. And if you happen to suffer from a genetic condition through no fault of your own, let it be known that there are many scientists out there working very hard to develop safe and effective cures that target those conditions. So I would say we're on the forefront of a revolution in cures, particularly as we add data science and new ways of de-risking the costs and the side effects of bringing better therapies to market that target people with known conditions.
I have a question from Amina. What do you guys think will be the next limit hurdle that genomics will face, if there is one? Significant technological, scientific hurdle. What are the limitations now?
I think each of us would give maybe different or overlapping answers. I would just quickly rattle off a couple. I would actually say one of the biggest challenges we have in genomics now, which I know Carlos will agree with me on, is that we're victims of our own success. We've had so much technological innovation that we generate far more data than we know how to analyze. We simply do not have we don't have a big enough community or powerful enough computers to analyze all the data in ways that we can dream about. So we are trying to play catch up on data science to keep up with our incredible innovations in genomic technologies. At a more societal level, I think one of the challenges, and we are keeping a close eye on this, certainly in my institute, is to make sure that genomic benefits everybody, that we don't just benefit a few, and we don't want to have, at the end of the day, genomics find its way into medicine and have it exacerbate health disparities. And there's a long history of medical innovations that have just widened the disparities gap, and we are hoping that won't happen with genomics, and we're working very hard at that at multiple different levels.
I would like to add something that follows to what I also presented earlier, and that's the importance of scientific user facilities for biological applications. Even though there's been a lot of progress in terms of figuring out the systems that would give us the three-dimensional structure of proteins, for example, there's still a crying need for facilities of the scale that physicists have enjoyed over the last century and still enjoy in proportion much greater than what biology deserves to have. It's not still something that biologists have embraced, are comfortable with respect to these facilities. But until and unless we do something like that, we will not be able to get to the next stage. And moreover, we may have difficulties anticipating and mitigating some of the dangers that were implicit in the question that was raised. Thank you.
And
Yeah, I would say that on the science frontier, I think that understanding how we create better diagnostic and prognostic tools is still a challenge, right? It's easy, quote unquote, to sequence. It's hard to interpret and make use of this in the context of care. And I'm optimistic about adding additional biomarkers. I think that's definitely going to help. And I would also agree with Eric that the big challenges are analytical and cultural, right? We actually don't have enough genetic counselors. We don't have enough and the emphasis on genetic counseling is on the counseling side, not so much on the genetic side. We need people with the ability to translate this to care providers, and I work a lot now with physicians in trying to get good diagnostics in their hands, and they want to make use of it, but they don't. They literally it's unclear how. If you trained as a physician 20 years ago, how do you make use of a whole genome sequence in your practice? That's just something that wasn't even imaginable back then, and so we have a lot of knowledge inequality that we need to address, and I think genomics, suffers from that, right? It should be something that helps us break down barriers and access to care, not accelerates and exacerbates disparities. And so I think those are all challenges that we're going to be facing as we deploy more and more into clinical enterprise.
And that, by the way, addresses, we could quickly, I see the next question coming in from Vicky, is artificial intelligence helpful to the field of genomics? Absolutely. And we're just scratching the surface. Of course, we're just scratching the surface how artificial intelligence is going to be used in many other applications as well. But I am quite convinced AI will play a big, important role in helping us dig out of this data challenge that we currently face.
Right. I agree.
Yeah.
I have a question I will follow up on. How has the approach been to sequencing the genomes of all the other species? Is it any cheaper now that you've done humans? Is there what is the systematization or method for it? Has it been abandoned? How's that being done?
Well, absolutely. The reduction in cost of sequencing DNA applies to humans as much as it applies to any life form, of any vertebrate, any invertebrate, any microbe, fungus, bacteria, viruses, et cetera, et cetera. So, yeah, I mean, there are systematic efforts going on. There's under various different titles, Zoonomia, Tree of Life, various big programs to sequence, all organisms on the planet, or all the organisms in a country, or all the organisms on every node of the evolutionary tree. And it's a whole field called comparative genomics, and it is spectacularly exciting. And that includes both organisms that are living now and those that are extinct. Because, of course, now we can also sequence out of fossils and out of ancient DNA materials, previous life forms.
Yeah, it's exploding.
Exploding.
How many species? How many species have been sequenced by now?
Well, it depends if you're asking about vertebrates. I don't know the number, but I mean, there are attempts to catalog these things, but it depends. I mean, if you're talking about bacterial species, I mean, it's thousands of different bacteria. Yeah. Among vertebrates, I think we're up to, I don't know, we must be at 1,000 or something. And also,
there's a genome 10K project, which is trying to do 10,000 vertebrates. I think Illumina just published most of the primates, so there's about 250 primate species. All of those have pretty much been sequenced. They don't have a draft. They don't have their own de novo assembly, but they've pretty much been humanized in terms of their use for interpreting human genome. And I'll add my second favorite species after humans, which is the dog, where we've been able to do lots of fascinating dog genetics. Again, there's a great dog consumer genetics set of activities. And in fact, one of my favorite papers, a paper we published with one of our colleagues at NHGRI, Elaine Ostrander, which was the insulin growth factor 1 gene and its role in body size. A company has now gone to market and is in trials with an IGF-1 gene replacement that would extend lifespan in dogs based on attenuation of insulin growth factor 1, because we know there's a negative correlation between the dog body size and how long they live. So if IGF-1 is implicated and you could reduce the expression of IGF-1, could you extend the life of your dog? So even very practical application of this into precision health that could someday see its way into humans as well.
How much are scientists now using simple tools that let them use the results of the Human Genomics Project without actually knowing how it works? You know, the way people use a personal computer, they don't know how the computer works. And in fact, are you getting a generation of scientists who are using it but don't know it?
If I understand the question, I guarantee you, I don't understand how GPSs work. You know, I don't understand how any of these things I do that tell me how to drive my car from place to place. I don't know how that works, but I use it all the time.
Right.
I know there are lots of physicians and scientists who regularly go to browsers that are basically Google Maps of the human genome, and they zoom in and they zoom out, just like you would zoom in and you would zoom out to find out where is a hotel relative to a gas station, relative to a convention center. But they're zooming in and out and saying, where is a gene relative to the element that turns that gene on or off? And they're zooming in and saying, and figuring out if that piece of DNA gets made into RNA. They are using that information very proud. They have no idea necessarily how that information got annotated into that Google map of the human genome. So I think the answer is yes. Some know it really well, but a lot of them are just consumers of that information, just like I'm consumers of GPS information.
Correct.
And you're not worried about this. Are there downsides?
No, I'm not. Well, I mean, there's always buyer beware. I mean, the internet, you know, you shouldn't just believe everything you read on the internet. And as a scientist, you're always skeptical of anybody else's data, right? So if you believe you do an analysis on the internet that tells you something about a region of the genome that you're interested in studying, you're going to make a biological inference. I am sure a good scientist is going to want to confirm it independently. But that doesn't mean that they need to figure out how it was originally found or how it was. I mean, they probably could read about it. But sure, they need to make sure they confirm their findings. But no, I think this is the whole thing. What genomics has done and what these Google Maps of the human genome, as I refer to it, is it raises everybody's boat. Everybody's now knowledgeable, so that what used to be an experiment that would take a year to perform is now a quick computer lookup and gives you the answer.
Anybody else want to address that? All right.
Right.
Yeah. How about the basics of making sure that the data is still available? How do you work to make sure that the hardware and the software is regularly upgraded and that none of the data is lost in older technology generations?
So that's a great question. It's something that I spend a lot of my time professionally working on, because in many ways people look. I can't answer it for all forms of, but for the National Institute for Health, the data that is being used by our scientists, we have to spend a lot of money storing this data, making it available, thinking about in the long run how it's going. The big transformation going on right now, because we have so much data that's generated, people can't store it at their universities. They can't it's just, so that's why all this thing around cloud computing has happened. And that creates opportunities, but also challenges. And the idea is, that how do you, and meanwhile, it's not so much, it requires money to maintain these databases. And NIH spends millions and millions of dollars every year to maintain them. But this gets used worldwide by scientists everywhere, and enables scientists, including in low resource countries, to do research, because maybe they don't have a laboratory, but they may have a laptop computer. And with a laptop computer, you can do an awful lot of powerful science in the cloud, using data that's available. So we are very attentive to data and the maintenance of data and making sure it's accessible and making sure that, with upgrades of software that you can still compute on that data. But it is an ever escalating challenge because the data keeps getting bigger and bigger.
Anyone else on that?
Well,
And actually, like,
from the NIH.
how about from the private point of view? I mean, Dr. Bustamante, how do private companies address that challenge?
Yeah, no, there's a huge ecosystem on cloud storage, and in some ways, right, the biggest businesses that have been built over the last decade have been data businesses. So if you think about AWS, it is the most profitable part of Amazon. When you look at Microsoft and its Azure platform, right, and the investment that it made in OpenAI, it was actually the largest cloud credit investment in history, right? It's billions of dollars of cloud credits that OpenAI got because that's what's valuable. So I would say, aside from storage, the other big piece of this is compute and building bigger models. So it's been fascinating that another, to bring this back to DOE and Ari's neck of the woods, quantum computing is back, and it's back harder than ever. And a big part of how that's being thought about is in these supercomputers of GPUs that have been pulled together so that they can do molecular dynamic simulations. And all of that is a testament to, not only aggregating the data, but more importantly, building models that can travel. And I'll add one last bit, which is that when genomics is at its best, it actually reduces the cost of acquiring the data to the point that storing it as DNA is often the most the most cost-effective way of doing it, and resequencing and making it a better draft. So, as to Eric's point, I've heard him say his kids will be still interpreting the human genome 50 to 75 years from now as we learn more and more. And I think that's true, right? It's a gift that keeps unwrapping as we add more and more technology on top.
I maintain my relationship with the Oak Ridge National Laboratory, that at least at this stage has the fastest computer. It's an exaflop computer. And I rejoice experiencing the amount of biologically related research that has a prominent position in the applications of the supercomputer. And even, discussions about quantum computers now include references to biological research and environmental research. Very, very comforting and reassuring.
So what is the international spread of genomics research? That is, I gather it starts, centrally in the United States, but how is it distributed around the world at the present moment?
I wouldn't give total credit to the U.S. I think we're among the leaders. Carlos, I think in his talk, talked about the UK Biobank. I would point out that the UK was major contributor to the Human Genome Project, as were about 3 or 4 other countries. So a good number of countries participated in the Human Genome Project. I think it has spread because it's everywhere in biology, and there's been a lot of capacity building efforts. I was instrumental in helping co-lead a program that the NIH funded with the Wellcome Trust, called the Human Heredity and Health in Africa, or H3 Africa Project, to try to get capacity building to bring the African continent up in having more genomics and genetics capability. Interestingly, the big emphasis there was on computational genomics because it's harder to have good laboratory infrastructure, but we found that we could really empower a lot of scientists in Africa to do experiments computationally. Along with what I was saying before. And then there's similar efforts going on in other places. I know Carlos has been involved in some in South America, Latin America, the Caribbean, and so forth. And it's an ever-growing effort because, again, we always are interested in global health and the sharing of health information and health technology. And we see genomics being intimately tied with future advances in medicine, and we want all human populations to benefit from it.
I, I guess, I mean, is it still though that you, I know that there's all these things, but can you talk about like what are the like the top half dozen countries where where genomics is done? I mean, can one make a list like that?
Um, sort of the countries that come to mind immediately. I'm going to leave some out, so I want my colleagues to help me. I mean, the U.S., the U.K., certainly Korea and Japan, China in a very big way, Australia in a very big way, Iceland you heard about. Certainly Germany and all the Scandinavian countries are doing all sorts of things in genomics. I'm probably leaving some out, but I think when you are able to reduce the cost of the most fundamental thing you do, which is sequencing DNA, when you've reduced it a millionfold, it makes it much more accessible to a wider spread of countries and the scientists in those countries. And so we have certainly seen the new sequencing technologies, which are incredibly cheap by comparison, be adopted more worldwide than what we were seeing during the Human Genome Project.
Thank you.
Yeah, I would add that the Middle East has made big inroads, especially in the last few years. Saudi Arabia, the UAE, they're all very, very interested in that space.
Yeah, I guarantee my list was incomplete. I just know it. If I thought about geography long enough, I'd come up with many other places, including Canada and others. Yes. Singapore is big time in genomics.
What is the education that you need now to go into genomics? That you were talking about how everybody, Dr. Green, came in from different fields. Is there a standard disciplinary? All right, there isn't. But what are the education skills one ought to be having for what the major entryways into genomics?
Well, I mean, there's a lot of different ways. It's about what you're going to do. I mean, the point is, anybody who's doing life sciences research is going to touch genomics, and so there's going to be sort of a minimal competency. I mean, you could ask the question, what's the minimal computer skills do you need to be a member of a modern society? I mean, you need to have some basic computer skills if you're going to do it. I think if you're going to touch life sciences, you're going to need some minimal genomic skills. You may never sequence DNA, but you're going to need to know how to access data about it. Or maybe you're going to be doing a lot of sequencing of DNA, then you need to know what's under the hood of some of these instruments. So it's no longer an isolated little piece of the scientific ecosystem, but rather it's pervasive. It's everywhere. So I can't think of any life scientist who's not in some way having some minimal competency in genomics, and that is why some of the fundamentals are being taught even at the junior high and high school level.
Well, actually, Dr. Bustamante, since you did come into this more recently, I mean, what was your educational pathway into the field?
Yeah, so I was a biologist very interested in genomics. So I ended up learning a lot of statistics along the way. So I got a master's in statistics at the same time I was doing my PhD. And it turned out to be fortuitous. My first job was teaching statistics at Cornell. So today I would say it's a good combination of computational prowess, and more on the data analytic side necessarily than necessarily on the other parts of coding. Although that's, if you're good at hardware, genomics is always going to be a gift that keeps on giving, because there are lots of interesting problems at that intersection. And I would say the other part is a curiosity about biology and medicine, right? Because ultimately it's a tool. Genomics is a set of tools that can be applied to a general class of problems, and so biology is rife with very smart physicists that come in and do physics by other means, right? So population genetics is the thermodynamics and statistical mechanics of genes in a population, but ultimately you need to know what direction you want to take that, and so you have to be question driven. So you've got curiosity about conservation genomics, about medicine, about agriculture. There's lots of really important, to Ari's point, problems of human significance where I hope genomics can play a role in illuminating solutions.
As I said earlier, I think there's still a crying need of scientific user facilities worldwide that can essentially reduce the burden of repetitive work that genomic scientists still unfortunately have to do, and to mimic essentially what we've learned from the big accelerators and these machines that have been used to probe the nature of the universe and so on. And there's still many such facilities that need to be funded adequately so that individuals don't have to repeat many things and have to relearn many things while they're pursuing their goals.
By the way, David, I see in the chat that Vicky asked another good question. I'm happy to take it. Chad said, she says, has the term junk DNA been discredited now? The short answer is, I think, yes.
Yes.
The word junk DNA got sort of that name because of the naive view that the greatest amount of biological information, perhaps almost all biological information, was going to be in the genes themselves, which only represents, we now know, only about 1.5% of all the letters of the human genome. And it was used in sort of a view that the rest of it must be junk. That is incredibly naive. And what we now have learned 20 years into having the genome in front of us, the genome sequence in front of us, and interpret it, is that indeed the gene repertoire of humans is actually not terribly impressive in numbers. We don't have that many more genes than flies and worms. And in fact, we have fewer genes than most of the things that you eat in your salad at dinner each night. And in fact, our complexity, and we believe that most mammals' complexity and higher organisms reside outside the genes in all the circuits and all the switches that dictate when, where, and how much genes get turned on and off. Our biological complexity is in outside the genes in what used to be called junk DNA. And it turns out to probably be what will be the hardest, most sophisticated part of the human genome to interpret. And it turns out to be inevitably going to be incredibly important medically. There will reside a lot of DNA changes out in our, what was once called junk, that turns out to be medically very, very relevant. And so I think by and large, it's been discredited. Now, does it mean that every single base in the human genome is important? Could you live without a base here or a base? Absolutely no. There's a lot that's not important. But this global labeling of major segments of DNA as junk is an incredibly naive phrase that has largely been discredited.
And these people that use that term regret it to a great extent.
Yeah. It's another example where people hated the Genome Project. Now they love, they've admitted, all right, bad term, bad idea, bad attitude. We win them over. We just do it one battle at a time.
Correct.
I will close with a science fiction question. Let us say that the Mars rover does find some equivalent of a billion-year-old Martian bacteria whose biological bits are there enough to be reassembled. And it's obviously it's not earthly DNA. One, how much of an advantage would you already have in trying to figure out how to, decode this, strange alien, beastie? And two, have you already been talking with NASA about this?
Go ahead, Ari, you may know more about it.
They have no question that, at NASA and several other places, this is something that enjoys considerable interest on the part of both serious and not so serious people, obviously because of the sensational aspects of such a case. So there's quite a bit of thinking going on, as far as I know. Whether we have to wait and see whether something like that happens or not, and until that time comes, we can only speculate.
And my immediate reaction is if somebody brought me back a specimen from Mars and they think it's a life form, the first discipline I would want to talk to would be chemists. Because the first thing you got to figure out is what is the information molecule in this living system? Is it a DNA molecule, or if it's not, is it another kind of nucleic acid? And what are the building blocks? Everything about genomics is built around the DNA molecule that we understand here on Earth. We understand the 4 chemical building blocks. We understand how to elucidate their order. And if you're dealing with a different blueprint molecule, right, first thing we need is a chemist to figure out what that molecule is made out of. and how it works. I think going through it a second time, if it all mimics what we do with DNA, we'll be ahead of the curve. But I want to start with basic chemistry.
Good for you.
Thank you. Dr. Bustamante, do you want to add to that?
Yeah, so I was able to be part of a really interesting project years ago where we sequenced a very interesting sample from the Atacama Desert that was thought to potentially be, extraterrestrial life. It was a very small humanoid, and we called it the alien mummy baby inside the lab. Turned out to be human. Turned out to have genetic defects that you could figure out from the sequencing, and weirder things. It's like the Twilight Zone. Weirder tales have been told, but I do think that if it's a carbon-based life form that has nucleic acids, we'll be able to sequence it on Earth, if not using the current Illumina technology, probably using PacBio or something else. So. And I think scientists are very interested in this, right? We remain, I think to the remain curious, and as they say, it would be an obviously waste of space if we're alone out there in the universe. So,
I mean, Earth reflects one experimental outcome, and it's not
right.
the best experimental outcome. You can watch the news lately, you can conclude it's not the best experimental outcome. There may be other life forms out there that are have better experimental outcomes.
Indeed.
Absolutely.
Dr. Pangloss will have words with you. I will, at this point, ask if you'd be so kind as to have some closing statements, each of you, and I guess in the same order, 30 to 60 seconds, and then I'll just wrap up for NAS.
Sure. First thing I'd say is thanks for hosting us today. I think we all love the opportunity to talk about not only the history that we've lived or the scientific contributions that we've made, but also to be able to discuss the exciting future that all of us see. It's truly something that when we were involved with the project, we knew this was historic, and we knew there'd be people like Carlos Bustamante who'd pick up the baton and do incredible things. And one of the most gratifying things of being involved in the Genome Project and now being part of the field basically since its inception is seeing all the different ways this has been impacting science across many different horizons. And as a physician, what I'm particularly excited about is seeing how this is finding its way into medicine and really, I believe, going to change the practice of medicine going forward.
Well, I thank the organizers for the invitation. I very much enjoyed meeting my good friends again, Eric and Carlos, that I've known over the years, and see that they're still working hard, and I applaud that. I try to still work a little bit here and there, but certainly not at the intensity of the years and the times of the Human Genome Project. On some issues, I mean, I will, like a broken record, go back to the importance of scientific user facilities for biologists and environmental scientists. These facilities are important for the health of the research endeavor and the kind of problems we face as humanity in terms of environmental problems, especially, and particularly the problem of global climate change, will require and demand the type of breakthroughs that only a significant investment in basic research and applied research will deliver it. Thanks again.
I want to echo my colleagues. It's always great to see Eric and Ari, and I learn so much every time I hear you guys speak. And look, I'm reminded of the quip popularized by Sir Isaac Newton, right? That if I can see further than others, it's because I stand on the shoulders of giants. So none of the careers that many of us dreamed of would have been possible without the pioneering work of the Human Genome Project. To me, it is the testament to unbelievable grit and determination and selflessness of a generation of scientists that made it possible well before at the dawn of the internet, at the time when we're just automating sequencing. And I feel really grateful to have been able to to carry a little pebble on top of that giant hill that got built by these guys. So thank you guys. Thank you, Ari, for all the work you did to get this funded. Thank you, Eric, for continuing to carry the torch. And there's a generation or two of scientists that are very grateful for that work because it was really unbelievably important and continues to bear fruit. I think that's the other message. So thank you
are
all.
most gracious. Thank you.
Look, and thank you all so much. So I'll just close with, so thank you. Thank you to our panelists. Thank you to our audience. Our audience, we do it for you. We can't do it without you. The questions alone are, they make this happen. So thank you to everyone. I will just do, let's see. Hi, if you enjoyed what we're doing, consider joining the National Association of Scholars or making a donation. We accept gold bars, silver too. Also, we do webinar series regularly. This closes our American Innovation, American Invention series, and I urge everybody to go back and look at the entire series, which is, I think, it was a great one. I've had a lot of fun moderating a fair number of them myself, but please continue to look at our webinars, both this series and others that we do. I will just repeat again. If you have questions you still want to have answered, send them to me, Randall at nas.org, RANDALL at nas.org. And again, just look for this on our YouTube channel, National Association of Scholars YouTube. Within 24 hours, you will be able to see, and hear all this immediately and in perpetuity. So, and I think that covers it. Again, thank you all so very much.
Bye-bye.